We just want to say thank you to everyone for coming. We're the mums that organised it, along with a few of the other mums who are scattered around as well. But thanks for coming. We're going to start the walk now, so we're going to walk that way. It just goes down to the end and then back.
In the sort of 6 or 7 years since we started, a big shout out to Deb and Lisa. Thank you for all organising. It's been amazing, and for turning on the weather. It's going to be partly rainy yesterday, rain tomorrow, but perfect today. So one of the things that the foundation does is try to find our most talented scientists in Australia and get them interested in working with Prader-Willi. And in Victoria, we were really lucky to do that with Professor Blewett. Professor Blewett was not working on Prader-Willi, was not even in her sights when we had contacted her. And then, by luck, she came across, when she was working on muscular dystrophy, she came across the action that activated part of the silent genes in Prader-Willi. And since then, she's gone on to, we spent $15,000 with her in the beginning, just piloting whether this is actually real, what she stumbled across. And since then, we've managed to work together to come up with $1.5 million to spend on her program of work. And we're hoping that that will be going to medicinal chemistry next year. So what that means is we've taken an idea, the shell of an idea that we wanted to find a gene therapy, through to actually identifying a mechanism and then identifying potential drugs or potential molecules that pharmaceutical companies can turn into drugs. So really crossing our fingers that that's going to take us a long way further. And then, so last year our latest grant went to Dr. Laura Layton from the University of Queensland and the Queensland Brain Institute. And I'm going to hand over to her to let her tell you a little bit about the work she's doing as well. Well, we're hoping that that this will be the beginning of another long partnership like we've had with Professor Blewett. Thanks, everyone.
Thank you so much, everyone. It's absolutely fantastic to be here today. This is amazing. This is such a great turnout. I'm just absolutely blown away. So I've been asked to tell you guys a little bit about what my research project is about and what we're kind of hoping to achieve in the context of Prader-Willi syndrome. So to put it in context a bit, I want to go right back to the basics and sort of talk about some of the building blocks of life. So when people talk about genetics, they're referring to DNA, the genetic code. And you probably know that every cell in your body has a copy of your complete DNA, and it contains all of the instructions for making and running you. And on the really tiny little molecular scale that we're talking about, that's a lot of information. That takes up a lot of space. So it has to stay safely in the cell's nucleus. And I kind of think of that as like having some big recipe books up on a high shelf in my kitchen somewhere. They're big and they're heavy and I don't want to damage them, so they stay up on the shelf. And when I'm cooking, I might have photocopies of a few recipes floating around in the kitchen. And the cell does that too. It makes some temporary copies of parts of its DNA using a related molecule called RNA. And just like photocopies, the cell can make as few or as many copies as it needs, and they're heaps smaller, so they can be sent around to all different parts of the cell. And most of those copies contain instructions for making a protein. And these are like the building blocks and the tools of the cells. So it's proteins that give your cells and tissues structure. It's proteins that digest your food, that carry oxygen in your blood, that pass the electrical signals around in your brain. And so this is kind of the flow of information around the cell. Is one of the main purposes of RNA is to carry the recipe for how to make a certain protein. But some RNAs carry other kinds of information as well, like information about how much of a protein to make or where in the cell something needs to be sent. So at the risk of overextending the metaphor, these RNAs are like post-it notes in the pages of the book and on the photocopies, giving additional context or making adjustments. And I'm working in the lab of an excellent scientist, Tim Brady, and our lab is interested in the dog ears, the highlighter pen, the post-it notes, all these extra bits of information that allow cells to precisely control how and where their genetic information is used. So I'm working on something called snow RNAs. And these are little bits of RNA that match up with the messenger RNAs that I was talking about before. And it's the same base pairing mechanism that sticks the DNA double helix together. It's kind of like a zip. And when it finds its matching RNA, it causes a chemical tag called methylation to be made to that matching RNA. And that can affect its properties quite a lot. It can change its three-dimensional structure, which really matters a lot for RNA, or it can make that message last a lot longer in the cell before it naturally breaks down and gets replaced. And so we care about this in the context of Prader-Willi syndrome because the critical region on chromosome 15 has a bunch of snow RNA in it. And people who have Prader-Willi syndrome are missing these snow RNAs. And at the moment, we don't know very much about how that might contribute to their symptoms. And compared to other snow RNAs, the ones associated with Prader-Willi syndrome are quite unusual. Most snow RNAs are in every cell in the body all the time, and they modify structural RNAs. But the snow RNAs from the Prader-Willi syndrome region are mostly just in the brain, and we don't know what other RNAs they're interacting with and potentially targeting with that modification that I was talking about before. And there's one called SNORD116 that we're really interested in this one in particular because different people with Prader-Willi syndrome can have more or less of that region on chromosome 15 missing, but the people with the smallest deletions that are known to cause Prader-Willi syndrome who have this, they're just missing SNORD116 and almost nothing else. And if you delete this gene from a mouse, you actually get a mouse that has a lot of the same features like increased appetite and sleep disturbances. And so we really want to find out what the snow RNA does. And so what I'm working on at the moment is mapping the interactions between snow RNAs and other RNAs. And I was saying before how they stick together like a zipper. And we get a chemical that goes in between the teeth of the zipper and sticks them together. And we can kind of capture these stuck zippers and sequence them and hopefully use that information to find out what targets the snow RNAs are physically interacting with. And then I'm hoping to search those targets for the little chemical mark that's left by the snow RNA. So because the Prader-Willi associated snow RNAs are so different from other snow RNAs, we don't know for sure if they even work in the same way. And so that's what we really want to find out with this project. So at this stage, what we're working on is a pilot project. It's foundational research. We want to find out what this is and how it works in a mouse. And we're really, really hopeful that the long-term results of this research might be the identification of new genes that contribute to Prader-Willi syndrome that we don't know about yet because they're in some other part of the genome and not in that critical region on chromosome 15. And so depending on what we find, it's possible that medicines might exist that already target those genes and pathways and that might be helpful for people with Prader-Willi syndrome. It's also possible that we might find new targets for drug development. So these outcomes are a fair way off right now, but science always has to start from a place of understanding and a place of hope. And so right now that's where we're at, and we're just so, so grateful for your support, and we really hope that we can make something happen here. So thank you everyone so much.
And thanks, Debbie and Lisa. Look, Finding 15 is all about the intersection of science and love. And we're just really grateful that everybody's come today and supported all the work we're doing for our kids. Thank you. Thanks, Matt.